welcome to our video on vaccine development and the COVID-19 vaccines, where we're going to be picking up where we left off from our previous video and talking about the two main features of vaccine development, one being safety and two being effectiveness versus efficacy versus efficiency of developing a vaccine. Within these two components, we'll split off and talk about vaccine clinical trials. We'll go through the different phases involved, one being your preclinical phase, two being your phases 1, 2, and 3, and last being your post-marketing phase. Then we'll go on and talk about a vaccine's journey. We'll hit on the points of its manufacturing, distribution, and quality control. We'll use all of this in relation to COVAX as our example, and we'll then go on to compare the different COVID-19 vaccines that are out there today. Lastly, we'll finish up with the different mechanisms of actions of all the vaccines available. First up, we'll go through the different components needed for vaccine production. This is something we touched on briefly in our previous video. We'll go ahead and expand on these two concepts. Very important. Number one is to focus on safety of the vaccine. And we do this really in three different ways. First, we test all vaccines preliminarily on animal models. If these prove to be safe, we then move forward to our human volunteers. These will be clinical trial participants. First and foremost, they'll be young and healthy, and we'll use this sample of population to compare to a control group who has not received the vaccine. And so from here, once safety is checked off, we need to consider evaluating the process of this vaccine. And this can objectively be done via effectiveness, efficacy, and efficiency. These three words can prove to be a little confusing. I'll start with E, and all have slightly different variation in definition. So let's go through and define each. Number one, what does it mean for a vaccine to be effective? This refers to the ability of the vaccine to produce the intended results in the field. Now, this is different from efficacy, right? Effectiveness is not the same. What does it mean for a vaccine to be efficacious? This refers to the ability of the vaccine to produce the intended results under ideal conditions. And again, this is slightly different from efficiency, which is the third E. So what does it mean for a vaccine to be efficient? This is referring to its ability to produce the intended results with a minimum expenditure of both time and resources. Next up, we'll be discussing how our vaccines developed. So most vaccines have been in use for decades, with millions of people receiving them safely every year. And as with all medicines, every vaccine must go through extensive and rigorous testing to ensure it is safe before it can be introduced. This is achieved through vaccine clinical trials, with 5 different phases that we'll go through and talk about now: our preclinical, phases 1, 2, and 3, and our post-marketing phase. So starting off with our preclinical phase, this is done without testing on humans, where an experimental vaccine is first tested in animals to evaluate its safety and potential to prevent disease. If the vaccine triggers an immune response, it is then tested in human clinical trials in 3 phases, with phase 1 involving a small sample of the population to confirm safety and the generation of an immune response while determining the correct dosage. Whereas phase 2 will employ a moderate-sized sample of the human population, and we will add a comparison group in which A select sample of volunteers will not receive the vaccine. Next, we have phase 3, where a larger sample will be employed, and this will be done to determine if the vaccine is effective against the disease it is designed to protect against. Whereas lastly, our post-marketing phase will assess both safety and effectiveness. Next, we'll go through the different steps of the vaccine's journey. I'm going to pull up an image here from the World Health Organization, where all of these steps are outlined. The first of which we just covered on the development and clinical trials. We'll then go through the pre-qualification step, followed by manufacturing, storage, delivery, and lastly, quality control. So I'm just going to move this image to the right so that we can break down each one of these 5 steps further. So first up, how is the vaccine approved for production? So once it has reached pre-approval stage following the clinical trials, the vaccine is assessed by the relevant regulatory bodies such as the FDA for compliance with quality, safety, and efficacy. Following approval, manufacturers can submit a vaccine to the World Health Organization for pre-qualification. This is another assessment process to ensure quality, safety, and efficacy. During global health emergencies, the WHO Emergency Use Listing Procedure, this is abbreviated as EUL, may also be used to allow emergency use of the vaccine. A current example of this being the global pandemic due to COVID-19. So next up, we'll hit on the manufacturing of the vaccine. Typically, companies will work independently to complete clinical development plans for a certain vaccine. Once it is authorized, manufacturing begins to scale up. The antigen is weakened or deactivated, and to form the full vaccine, all ingredients are combined. The whole process from preclinical trial to manufacture can sometimes take over a decade to complete, unless a global pandemic, as the current COVID-19 one, prompts all researchers and developers to work together on several different phases in parallel to speed up results. So next up, we'll talk about how is a vaccine stored. When a vaccine is too hot or too cold, it becomes less effective or even inactive. If stored at the incorrect temperature, vaccines can be ruined or unsafe for use. Most vaccines require refrigerated storage at between 2 and 8 degrees Celsius. Some require temperatures as cold as -20 degrees Celsius, with the newer vaccines needing to be kept ultra cold at -70 degrees Celsius. Next, we'll go through how is a vaccine shipped. So to maintain this cold chain, vaccines are shipped using specialized equipment that does not compromise the integrity of the product. Once shipments land in the destination country, refrigerated lorries transport the vaccines from the airport to the warehouse cold room. From there, ice boxes are used to transport vaccines from the cold room to regional centers where they're stored in specialized medical-grade refrigerators. And lastly, we'll touch briefly on quality control. This is once vaccines start being administered, national authorities and the World Health Organization constantly monitor for and possibly establish the severity of any possible adverse side effects and responses from people who have received the vaccine. Studies are often conducted to determine how long a given vaccine remains protected. So now let's take everything we've talked about so far and discuss it in terms of COVAX. So what exactly is COVAX? COVAX was created in response to the global COVID-19 pandemic, and with more than 2/3 of the world engaged, it is the largest and most diverse portfolio of COVID-19 vaccines, and as such represents the world's best hope of bringing the acute phase of the current pandemic to an end. And so let's look at what this means and use the US as an example of this. A total of 6 companies have made deals with the US government for a total of 800 million COVID-19 vaccines. we'll break down these 6 companies a little bit further down. We'll go through each and we'll talk about a timeline of the vaccine distribution. So first up, we have the Pfizer-BioNTech company, who's agreed to provide 100 million vaccines to the US, followed by Moderna with another 100 million. Then a little later, we'll have AstraZeneca with 300 million vaccines, Johnson & Johnson with another 100, and finally we'll have Novavax and Sanofi with each contributing 100 million vaccines. So kind of grouping these companies together, we'll see that the Pfizer, BioNTech, and Moderna companies were both ready to distribute their vaccines as of December 2020. This is when they both got immediate emergency approval from the FDA, the World Health Organization. AstraZeneca, Johnson & Johnson will not have theirs until February 2021, and Novavax and Sanofi will be ready in April 2021. And to further break down this timeline in terms of who exactly will be vaccinated to achieve herd immunity, looking at March 2021, by then we should have essential workers and high-risk individuals, to include people over age 65, vaccinated. Whereas by May 2021, we will be starting to have all people over age 65, regardless of high-risk conditions, as well as the general population starting to be vaccinated, in hopes of achieving at least 70% of the U.S. population being vaccinated by October 2021 to achieve herd immunity. And so currently there are 2 vaccines available in the U.S., the Pfizer-BioNTech vaccine and the Moderna vaccine. As such, we'll go through on this slide and break down some similarities and differences between these 2 COVID-19 vaccines. So starting with our Pfizer-BioNTech vaccine, it has proven to be about 95% effective when administered in 2 doses, 21 days apart, of about 30 micrograms, whereas Moderna has been administered 28 days apart at 100 micrograms. These are stored in 10-dose vials, whereas Pfizer is stored in 5-dose vials. The Pfizer vaccine does require some dilution, Moderna does not. They both had about the same amount of trial participants, and most common reaction has been injection site pain. So those were some differences, with the biggest one probably being the cold storage, where Pfizer really needs an ultra cold environment, whereas Moderna has a little bit more leeway there. On this slide, we'll go through and talk about the similarities between these vaccines, in that they are both mRNA vaccines. We'll define what this is, we'll get some facts straight, lots of myths debunked, and then lastly we'll go through an ingredients list. So COVID-19 mRNA vaccines, these provide instructions for our cells to make a harmless piece of what is called the spike protein. This is found on the surface of the virus that causes COVID-19. These vaccines are given in the upper arm muscle, and once the instructions from the mRNA are inside the muscle cells, they will follow the instructions and make the protein piece. After this is made, the cell breaks down the instructions and gets rid of them. Next, the cell displays the protein piece on its surface, and our immune system recognizes that the protein doesn't belong there and begins making antibodies. After developing antibodies, our immune system has now learned how to protect against future infection. And the benefit of this, like all vaccines, is those vaccinated gain this protection without ever having to risk the serious consequences of getting the real virus. Some facts regarding these mRNA vaccines is that they cannot give someone COVID-19. Again, these vaccines do not use the live virus that causes COVID. Additionally, they do not affect and or interact with our DNA, because the mRNA never actually enters the nucleus of our cell where the DNA is made. And lastly, there has been complete transparency around the ingredients used in these vaccines, and you can find the fact sheet posted online as well as referenced in my textbook, where the link is in the description of this video. And lastly, of note, mRNA technology has been around more than 20 years now and researched amongst various viruses to include the Middle Eastern Respiratory Syndrome, Zika virus, even the rabies vaccine. Early stage clinical trials were found to be safe and efficacious. However, funding quickly dried up, and with no funding, unfortunately, there is no scientific advancement. However, due to the recent global pandemic due to COVID-19, funding has been no issue. And with that said, with our next slide, we're going to go to our black screen and put together everything we've talked about in this video as well as our previous video, draw a picture of what the mechanism of action of these vaccines looks like. And what we want to see specifically is how these vaccines produce an immune response. And more specifically, how do they produce antibodies so that our body knows to fight off this infection in the future? So to start off, I'm going to be drawing the SARS-CoV-2 virus. This is the virus that causes COVID-19. And I'm drawing it out to show specifically these little yellow bubbles are what is known as the spike protein of the virus. We're going to abbreviate that as S protein. This is what has been shown to be very notorious in infecting and causing symptoms. So how these vaccines work is that they're taking the mRNA of this protein that codes for this protein, right, and kind of working backwards. They're placing it into this nanoparticle. So the orange circle I've drawn is a lipid bilayer. The little blue squiggly line, that is our mRNA. And so we're going to see how this is going to be incorporated into our cells via the vaccine to elicit an immune response. So for the red square, this is an example of one of our cells. I've just drawn the nucleus with a double-stranded DNA. The orange lipid bilayer is going to come and fuse with our cell, where the mRNA will then be able to enter. And as you can see, it is now in our cell. It does not interact with our DNA, never goes into the nucleus, never alters our own DNA. These next two red dots I've drawn, these are ribosomes. These are proteins found on your rough endoplasmic reticulum. That is what your mRNA is going to interact with. That is a process known as protein translation. So this gray dot is this new protein that is going to go and get expressed on the cell membrane. Those are the next two squares there that I'm drawing in pink and purple. These squares are both proteins that are expressed on our own cells. One is called MHC2 and the other is called MHC1. And we'll go through two different pathways now of how we're going to be mounting an immune response. The first pathway is going to involve the MHC2 complexes. These are going to be cells that are only found on a couple cells, 3 to be exact. These are known as antigen-presenting cells, APCs. Your B cells, your macrophages, and your dendritic cells. This is something we discussed in our previous video. So these MHC2 complexes are going to attract a type of immune cell known as a T helper cell that also has a membrane receptor, that's the square I just drew, known as a TCR complex that will come and interact with this new protein via its CD4 arm. Remember, CD4 are for T helper cells per our last video. This interaction activates this T helper cell that then releases a bunch of cytokines, interleukins 2, 4, 5, you name them, that then activate B cells. This is going to have a cascade effect. This is important because these B cells are then going to proliferate and make clonal B cells, plasma B cells. These are very important because their role is to form memory cells by making immunoglobulins. These are antibodies, often depicted as a Y shape. This is important because in the future, when you see the real virus, your body has already mounted a response. So now going back to our second pathway, that involves our MHC1 complexes. These are going to attract cytotoxic T cells, which have their own TCR membrane proteins. These have CD8 protein arms that will reach out and interact with this protein. And when these proteins interact, lots of dangerous immunomolecules are going to be released that are going to kill the cell, basically, at hand. Now, it is also important to note that these molecules will also act on the B cells in our first pathway, which will amplify our immune response so that there are even more cytokines being recruited, even more interleukins being recruited, so that more clonal cells, plasma cells are being formed, antibodies produced, immunoglobulins stored for memory. And now you have to remember, all of this is happening after you've gotten your injection of the vaccine. Because you are mounting your immune system, you're mounting this immune response, some people may experience some adverse effects, very mild, muscle aches, slight fever. This will only last a few hours while your body is preparing to defend itself against future infection. And with that said, this is an overview of